right, let's do influenza next. Interesting virus. So this cartoon is from 2009. This is from the outbreak of what was called the swine flu at the time, H1N1. And so they're walking along together, and Pooh Bear is thinking to himself, "Well, if this pig sneezes, he's dead." I like it. Anyway, yeah. Let's go and talk about influenza. So, I'm breaking this lecture up into a bunch of little parts. I think there's maybe going to be 7 parts, and each one of them is going to be in this format. So, a PowerPoint with nice talking to go with it. And let's learn all the parts. So, this is an influenza virus. And so, we got nice little pictures here. That's the purple thing up in the corner. It looks like 6 of them. They're in the group called the Orthomyxoviridae, and everything in the Orthomyxoviridae has negative sense RNA. So it's single-stranded, but negative sense. So this is pointing the opposite direction to what a coronavirus RNA was. And all that means is that you can't translate an influenza RNA to make protein directly. But what you've got to do is make a messenger RNA from that. So instead of packaging the messenger RNA, they're packaging the template that makes the messenger RNA. And so you can look at them as being one step ahead or one step back. Either they've already made the template and they're ready to go, or they're one step back and they still have to make a messenger RNA before anything happens. Regardless, you know what? It seems to work pretty well out there in the world. So there are 8 segments. There are currently 12 recognized proteins. I think there's one other one that's a version of one of those other proteins, so you could say 13. But this is also unusual. Whereas a coronavirus or HIV would be one single piece of RNA, the flu virus is actually broken up into 8 smaller pieces of RNA for type A and type B, and 7 pieces of RNA for type C, the flu C virus. So, similarly to coronaviruses and HIV, this is going to be an enveloped virus, which means it's going to have a membrane on the outside with proteins sticking out of it, and the membrane will be something that the virus took from its host on its way out. And so there are flu A and B and C. There is an influenza D now, but so far it's only in cows, so don't worry about it. And there are also lots of other related viruses in fish and lots of different kinds of primates. So chimpanzees and other monkeys, primates, all sorts of things like that seem to have viruses related to the flu virus. So influenza A virus, you'd think of that as the main human flu, and you'd be right. That's the one we vaccinate against every year, or one of the two. But really, type A is a virus of wild aquatic birds, and that's where it comes from, and that's where it goes back to. And so these are birds that migrate around the world, and the coolest thing is that they'll be up on a lake in, I don't know, Alaska or something like that, and they'll be eating little fish, and they will poop out flu virus. And then when that lake freezes, it can actually trap the virus. And even though the birds have migrated back down south, when they come back north to eat the fish again next year in the summer, that virus will actually thaw out and the virus still works. And so you can get them being reinfected, or they'll fly back with their children and then all their children get infected in this way. So yeah, that's fairly terrible. Influenza B is a virus that so far is only found in humans. And there are also seal versions of influenza B virus. This one is also pretty frequent, and we also vaccinate against this. So if you got the flu vaccine this year, you would have got a vaccine against 2 strains of influenza A. One's called an H1N1, and the other's H3N2, and against 2 strains of influenza B. And they have different names, but they don't have different H and N types. So yeah, just two B's and 2 A's. There's also influenza C. It's much rarer than A or B, and it only rarely causes people to have severe enough illness that they would have to go to the hospital. So it tends to be mild. B can actually be fairly severe, particularly in young children. So the flu will hit the very old and the very young the hardest. All right. So, what does a flu virus look like? Wow, here you go. Here's a neat little colorful picture. Yeah. So, it's going to be about the same size as a coronavirus, about 100 nanometers in diameter. It's actually closer to 80, 85, I think. But some of them turn into these long, sort of sausage shaped tubes. And so, yeah, it's a decent average size. Generally, they are spherical, so they're just round like a ball, but they can be a filament shaped, which would look like a tic tac if it's really short, up to a sausage if it's pretty long. And inside each one of these viruses, you have a little cluster, and it's going to have the RNA segments in there. And so you will have all 8 segments of RNA. And they're packed together in a particular way so that they make a little block of all 8, and they'll all have their top ends touching each other and their bottom ends touching each other. At the top end of each of those RNA segments, you're going to have a collection of 3 enzymes. So these are our first 3 flu virus enzymes, and they're going to be the polymerase basic 1, polymerase basic 2, and polymerase acidic. And so these are the 3 proteins that together make up the RNA polymerase of the flu virus. So this is what is going to copy the flu virus. So they start out with the template, the thing that they're going to make copies of, and they start out with polymerases already stacked on the end of it, ready to go. So as soon as this virus hits your cytoplasm, boom, it is going to start replicating, which is really neat. Actually, the virus is going to go into the nucleus to replicate, but theoretically it could once it got to the cytoplasm. It just chooses to go there so it can hide a little bit better. So that's kind of neat. Let's see. So we've got those 3 proteins on the inside for sure. On the outside, you've got 2 main proteins, and those are going to be the neuraminidase, which is called Na usually, and so those are the orange ones at the top and bottom. They look like little Thor's hammers. And you're going to have the hemagglutinin, or HA, which are the ones that look like little red ice cream cones sticking out of both sides of this thing. And so those are the 2 proteins that are going to do the job together that the envelope protein of HIV or the spike protein of coronavirus did. So these are going to bind to the cell receptor and cause fusion. And then neuraminidase is kind of a neat thing. Its job is to cut the virus loose from its receptor so that it can go off and infect somebody else. So the hemagglutinin is real important at the beginning of infection, and the neuraminidase is real important toward the end of infection in one cell. Now, in the membrane, you've got 2 other proteins. So let's look at those. You've got the matrix protein M, and oh my gosh, it does the same job as the matrix protein of HIV did. So it's going to bend the membrane like a little rainbow and lock together and make a nice little shield on the inside. And it's also going to bring together all the components. So it's going to bind to the neuraminidase and to the hemagglutinin. And it's going to hold on to those segments as well of RNA. And so also you're going to have M2, which is an ion channel. And so this is going to be pretty much identical to, or it's going to do the same job as the E protein of coronavirus. It's going to let little hydrogen ions into the inside of the virion when it's time to fuse. And so this is going to be how M knows to let go of everything so that we can get ready to unload this virus. And so it's going to let those H plus ions in, which means, you know how this is going to enter? Yeah, you can already see it, right? It's going to bind to a receptor on the surface. It's going to get taken in through a clathrin-coated pit. Once it's inside, we're going to acidify that vesicle, and then the hemagglutinin is going to go through some conformational changes. It's going to throw out a fusion peptide that looks like a little hook, and this is the one that's actually solved, and we actually know that it looks like a hook. And then, you betcha, it's got 2 heptad repeats, which are going to snap together just like they did in HIV, just like they did in coronavirus. And once again, it's going to go fishing, and it's going to try and catch a big one, trying to hook a cell and get into that cell. The last protein on the inside is going to be a thing called the nucleoprotein, and they haven't even really drawn the nucleoprotein there. They're just pointing to some of the strands of what I would take to be RNA. But the nucleoprotein is going to do the same job as the nucleoprotein of coronavirus. It's just a personal protector for the RNA strands. And it's also going to be picked up by the M protein, so the matrix protein. So, yeah, we've got different names for some of the components, but boy, do that, don't they sound familiar? I'm hoping that things are starting to sound familiar and really starting to click by now. I tried to pick out some viruses that work kind of similarly, despite being really different. So HIV starts out as RNA, turns into DNA. Coronavirus starts out as positive strand RNA and is all in one piece. And influenza virus starts out as negative strand RNA and 8 pieces. And you know what? Yeah, their virions are all quite similar. So that's good. Okay, real quickly, a little bit about disease. So, the symptoms of flu you probably already know. So, fever, chills, lethargy, which just means, you just don't feel like going anywhere. You just, want to lay down. Aches, particularly in the joints, but also sometimes headache. But yeah, muscle aches are very common. Coughing and sneezing. Yes, you know about the flu. And just sort of a general discomfort and pain all over. It's just miserable. It can lead to pneumonia, and in some cases, it can lead to death. Yeah, if it is not treated. But we do have some treatments for the flu. So yeah, so that's nice. A little better than for coronavirus. Shown down here at the bottom is the most famous and biggest flu outbreak. It was in 1918 and 1919. Minimum estimate is that it killed about 50 million people. I've seen estimates that are much higher than that, closer to 100 million. And the fact is, we don't know. This is before there were refrigerators like we have today to preserve the virus, like freezers. So we don't have any actual tissue samples from the people that were infected then. Although somebody went out to a graveyard in Alaska and managed to put this thing back together, which is cool, but that's going to be the subject of a future little bit of this. And so we really don't know if these people died from just the flu or like the flu plus bacteria. And also, they didn't have the same kind of medicines at the time as we have now. So it was really, really bad. But it's hard to say for sure whether or not the same virus introduced today would be as deadly. Yeah. Who knows? So I'm using a set of slides that I stole with permission from a colleague over in the UK. And the nice thing about the UK is that it is similar in size to, let's see, if you took Arkansas, Texas, Oklahoma, and Louisiana, put the populations together, and then doubled it. That'd be about the size of the population in the UK. And yeah, in general, we get the same virus at about the same time, so this is comparable. Shown over here, we've got lists, and you can see the number of viruses, and look how they peak every winter. This is a virus that comes back in the winter, and there are lots of people that will try to tell you why this happens, but the fact is, nobody really knows why this happens. Yeah, is that disturbing? Yeah, maybe. I don't know. Huh. And there are a lot of cases every single year, and the deaths also peak. So we think that in the U.S. anyway, or no, maybe it's in the world. No, I think it's just the U.S. We think there's somewhere between 10,000 and 30,000 deaths a year um, from influenza viruses. So it's really bad, but most of the people that are dying are the extremely young and the extremely old. So people that don't have an immune system yet, or people whose immune system has fallen apart with age. And so it's kind of understandable. This is picking on the weak. Yeah, the flu virus is a real bully, and I don't like it. Okay, so for example, in a given year, this is what 1 year's flu population happens to look like. So we had influenza A, H1N1, we had influenza A, H3N2, and we had influenza B, both types together. And you can see this was from a year where the vaccine against influenza A worked pretty well. So we pretty much knocked out the H1N1 and really knocked down the H3N2. So the H1N1, all these H and N numbers refer to the hemagglutinin and the neuraminidase. And there are several different types. I think there are You know what? We can look it up. We can look it up and get the exact number. There's something like 18 or 19 hemagglutinins and something like 12 or 13 neuraminases. The last time I checked, but they keep adding them as they find them. And so in this one, it shows that influenza B was actually causing more disease, but influenza A tends to be associated with more severe disease. Hi. So let's talk treatments and vaccines. We have the old treatments, things like M2 inhibitors. So M2, remember, forms a little channel. It gets four of the M2 proteins together. And when they're together, they can let those H plus ions go into the virion. And when they do that, they make the matrix protein M1 rather, let go from the nucleoprotein, NP. So that just helps the virus to get ready for fusion, and it's equivalent to the maturation process of HIV, except it is done without proteases. When the H plus gets in there, it just makes everything let go. Amantadine and rimantadine are two drugs that look like a little bathtub plug, and they just go in there and they plug that M2 channel. So that even though all the other steps happen, we can't get the inside ready, and essentially the virus stays in the immature form, and it cannot actually finish fusion. It can get to hemifusion, and it just gets stuck. So that's kind of neat, and that helps us beat the virus. Other possible inhibitors are things like Tamiflu and Relenza, and so you will have seen these or heard these talked about, I would think. The technical names are oseltamivir and zenamivir, and they are both things that bind to the neuraminidase, so that's the protein that's the N of H1N1, for example. There are also many different varieties of vaccines. The standard and best version of this is going to be a split vaccine. So it's going to have broken down virus and it's going to have two different influenza A strains. And the best vaccines that are available now actually have two different influenza B strains. Yeah, instead of the one that's listed there. But there are many different varieties, and there are some that are grown in eggs, some that are grown without eggs for people with an egg allergy. There are ones that are inactivated in different ways. There are live vaccines that'll give you a very mild version of the virus that will absolutely give you just great protection against getting the very bad form of the virus later. And there are versions of the vaccine that absolutely won't harm you. And these are very good for giving people who are old or already have fragile immune systems, something like that, just to give them a chance to help fight off the virus. So the most recent addition is these live attenuated vaccines where they'll actually squirt a little bit up your nose of a live flu virus. And for the first year or two that they tried this, this was just incredibly effective. You got sort of 90% effectiveness rates, as opposed to about 40% for the normal flu vaccine. So it was fantastic, but for reasons unknown, after the first year or two, the effectiveness kind of dropped off and it ended up about in the same ballpark as the inactivated vaccines. And so, if you can take an inactivated vaccine that has no chance of hurting you and it causes the same amount of immune response as getting a live virus put up your nose, then why do the live virus, right? Because there will be a few people that would have adverse effects. And so, for the most part, the world has switched back to the inactivated vaccine. But I think they're going to keep looking at this and trying to find ways to make it work because the effectiveness in that 1 year was just astronomical. Really good. All right, thanks. That's the end of part one.